Welcome to the Department of Renewable Resources seminar series for March 15th, 2012. Our speaker today is Dr. Nadir Erbilgin, Canada Research Chair in Forest Entomology and Assistant Professor in the Department of Renewable Resources at the University of Alberta. Today he will be speaking to us about invasion dynamics of the mountain pine beetle in jack pine forests, the roles of host tree resistance, competition, symbionts, and environment.
Okay, thank you very much. I'm not sure I should call this crowd as beetle-loving crowd, but we're going to talk about on this subject heavily. Just want to mention that even though I put environment in the title, but because of time constraints, I kick out. I just want to focus on host resistance, competition, and symbionts. And then this is the people that really do the most of the work. We're just sitting in our in the office and writing grants and preparing our teaching courses, but this is the group that in my lab particularly do most of the work pretty much, especially the one that I'm going to present today. This is sort of brief, very, very brief outline, what I'm going to talk about it. I know some of you might be familiar with this mountain pine beetle biology, but I just want to go through very briefly what it is. And then I want to introduce this concept called invasion biology. And then as then I want to switch to this invasion of jack pine forest by mountain pine beetle, and then talk about a little bit Conifer defenses. I'm just going to pick a little bit information. I will provide some little bit information on that. And then this subject is to come into we have some data generated in our lab with collaborators. I want to focus on these interspecies interactions, the role of microbial symbionts in the system, as well as our most recent finding about pheromone production of mountain pine beetle in jack pine forest. I think you're going to love it. That's really cool studies, all of them. And I want to summarize in one slide where I'm going with these studies in the next, well, I don't want to say next decade. Mountain pine beetle is a simple beetle. Like, we have thousands of beetle species in North America or Europe in the world, about 6,000-7,000 species, but only few of them really cause sort of economic concerns. But overall, they are cryptic insects, means that they spend pretty much their entire life under bark. And then, they attack the trees, they colonize it, as you've seen here, they lay eggs, the larvae emerge, they feed on, and then after that, beetle emerges and causing, in some cases, in this case, mountain pine beetle, causing huge epidemic that we've seen since 2001 in British Columbia and Alberta. And this is the very simple biology of that. This is specific to what we have in our region here, mountain pine beetle. They usually disperse from their host, adult beetles, around late July to early August. And then they find their host, they release aggregation pheromone, they colonize the host if everything works out well. And then they lay eggs. Eggs pretty much complete their development under bark. They spend winter as a larva, and then they pupate around late June and July, and we see this brood emergence from the same trees the next year. This process takes 1 year for mountain pine beetle, but some other species in southern U.S., for example, this is repeated about a couple of months, so you will see 5 or 6 generations of beetles. In this system, beetles, I want to mention that beetles have 2 population cycles we call endemic and epidemic. Endemic cycle means the population is really, really low, all right? And epidemic cycle, the population numbers are really huge. And in both situations, they have a different dynamic that I'm going to tease them out a little bit later. But normal endemic conditions, these are the major factors that pretty much keep beetle control. Host resistance are one of the most important one. As you see here, the beetle stuck on the resin. We have a lot of invertebrates, parasitoids, and predators that attacking and killing. Sometimes we see the pathogens, and some birds, woodpeckers, for example, they pack it. And then competition is really important, intra or interspecific, between species or different species. Competition and the weather, especially winter mortality, is really important. But in some cases, these organisms, they don't really do a good job, or other environment or other factors, due to environment or other factors, wow, you see a big mass of outbreak. This has happened in British Columbia, and this is the very small spot in 1999. This is what I have in 2007. I mean, 6 years, 7 years is really not much, but this quickly happened, and they destroy about 14 million hectares of pine forest. This is BC part, and cause very, very big sum of economic loss. And then scale and impact of mountain pine beetle is not predicted ever before. And then, well, Alberta is neighboring to the BC. We also shared some beetle epidemics. This is taken from the SRD website. This is 2005 where the beetle is, and 2011. This is the most current beetle population. Here is an Edmonton. It's really high. We've seen epidemic too. And the concern is, Alberta is a very unique place. We have a lodgepole pine forest, we have a pure lodgepole pine forest, we have a pure jack pine forest. In some places here in this part of northern Alberta, that those two species hybridizes. And then those hybrid areas is really important for sort of provide the transition point from one place, from lodgepole pine to the jack pine forest. And then sort of a breach, basically. The question is remaining here, well, what would happen in the jack pine, whether the beetles will be successful or not? That is the main questions. Different groups, different labs in the States, in Canada, they work on different aspects. But I usually mainly focus on, I assume that beetle will be successfully established in jack pine forest, but their population will be low. I really doubt we will see very large epidemic population for a while until the weather and other climatic factors might change in the near future. Now I want to introduce this invasion biology concept. That is the definitions, the ability of species to expand their current range. When you think about it, the invasion process is not really easy process. Insects or plants, they have to go through a very series of biological or abiological factors that they have, the blocks that they have to go through. When we're talking about the insect invasion biology, we have to really focus on the history of the plant-insect interaction, for example, whether they evolved together or this is recent evolution or is more long-term. We talk about insects, pretty much all our insects, including humans, humans are not insects, but overall, we all associated with the bacteria, all other microorganisms. They do a lot of works, and insects too. Especially for mountain pine beetle or bark beetles, they're associated with several group of organisms. I'm going to talk about it, but they do play a really important role. And the competition is really important. Within and between species competition, always important. We also plants. We're talking about the plants. They are not uniformly suitable for insects. Plant genetics are important. Adaptation, I call it. Adaptation means a reflection of genetics and environment, basically, interaction. And this part, the one that I'm usually focused on, resistance and tolerance mechanisms in plants. And then environment, when we say climate, temperature, precipitation, or physical environment, slopes and aspect sources, as well as soil resources, water. I think water is going to be very most influential factor affecting plant stress currently right now as well as in the future. Now I put them together, even though I'm an entomologist, but I try to focus on the plants. I try to look at things from the plant perspective. In many cases, because why do I focus on the plants? Because plants are really important in many ecosystems, and they develop a rich set of chemically mediated interactions. Think about it. If you think about it, the three species, from seedling to all the way to the maturity, they are subject to a lot of attacks from insects or diseases at the various stages. But both of them are surviving, means that both organisms are doing very well, plants and others. From this angle, I'm just going to focus on the plant resistance mechanisms, because resistance is really important to understand the plant responses to all other organisms. Because the plant resistance changes a lot due to environment, due to biotic factors. They changes a lot. Understanding those changes, what factors really govern those changes, will be very, very important. Just quickly, I'm just going to go through here, looking at the pine defenses against bark beetles. Again, I'm looking at the pine defenses from bark beetle perspective. We're talking about two types of defenses, mechanical and chemical. Mechanical defenses, again, these are providing toughness or thickness, making providing blocks for insect not to enter the tree. And this is the tree. We have the corkborg outside. We have the phloem, where the beetles usually feed on, bark beetles. We have the cambium. We have the sapwood and hardwood. This is pretty much it. And then the most important thing is actually plants have also chemical defenses. This, gotta tell you, under bark is a really nasty place to be. In many ways, it's difficult to get in. They have to deal with a lot of chemicals such as resin. This resin is very complex and contains a lot of insecticidal that harms insects or kill insects, or fungicidal compounds, or they kill the fungi basically. And they have to go through those things. In many cases, I can guarantee, even this is true for mountain pine beetle, most of the beetle attacks are unsuccessful when their numbers are low. But when their numbers are high, epidemic attacks are usually highly successful because they rely on the numbers. Anyway, these compounds, resin occurs as a pool of stored chemicals called secondary compounds, such as terpenoids and phenolics. Then they usually released upon attack and then try to make the life, give a lot of hard time to the beetles. In many cases, again, the beetles are stuck on the resin and they die. In other cases, they are very successful. And then just going to give a little bit more information about these defenses. When we're talking about plant defenses, we're talking about two levels. One of them is constitutive level defenses. The other one is the inducible defenses. Constitutive defenses, they are present before an attack. It's always there. And thought to be they are general and broad-based defenses. They repel or delay a lot of organisms, not targeted as single organisms. And then inducible defenses usually activated in response to the attack. And then, and they usually fine-tuned according to severity of the threat. And they are usually target-specific. Now, in my program, I try to focus on this invasion biology of mountain pine beetle. Again, I put the plant defenses in the system. And then plant defense is a reflection of a lot of factors. Okay, plant defenses really changes due to environment, the biotic and abiotic factors. In my view, that plant defenses, whether the plants are suitable or not to the mountain pine beetle, it really determines the beetle invasion success here. The plant defenses, as I mentioned, is a reflection of environment and resources, such as water and nutrients and the temperature. There is some direct interaction with this. We're also talking about a lot of organisms, not necessarily insects, a lot of diseases. But I put the invasive organisms here. This is the really novelty comes, when we're talking about plant-insect interaction, novelty really comes from the invasive organisms. They are not evolved together, but they have to go through those plant defenses. They have to overcome somehow. How do they do it? That's the subject that I'm going to emphasize today. When we're talking about insects or organisms, especially for herbivores, insects, they associated with the symbionts. And then this environment or resources, they also affect direct effect on organisms. That's really not the focus that I do. I look at the indirect interactions. When I say indirect interactions, those are the interactions usually go through the plant defenses. For example, environment might affect the plant, such as soil resources, such as drought, might affect the plant defenses, make them suitable or resistant. Those susceptible or resistant plants may affect the interaction with the organisms, native or invasive organisms. And this is the thing that I usually focus here. In between native organisms and invasive organisms, they also interact together indirectly by affecting the plant defenses. For example, if these native organisms make the plants susceptible, those susceptible plants will be highly desirable for invading organisms. This is truly the case for mountain pine beetle. When we look at the jack pine forest, wow, that's a big, big forest in boreal forest. We see a lot of organisms associated with it. A lot of diseases. We have armillaria disease, for example. We have a jack pine budworm. I single out jack pine budworm because it's one of the most important defoliators of jack pines. We have a twig beetles and insects attacking the top of the tree. We have a needle blights that attacking the needles. We have a sawflies that eating the needles of the jack pine. Many. And we also other soft acting diseases are called dwarf mistletoe. Why this is important? This is important because during the endemic cycle, mountain pine beetle cannot attack healthy trees. They intentionally seek the trees that they are susceptible or stressed or weakened. what are the most significant stress agents in boreal forest? And these are the organisms, usually, that stressing the plants. Therefore, in my view, those organisms that stressing these jack pine trees make them really make my mountain pine beetle colonization easy. Therefore, promote the invasion of the boreal forest. That is my logic. I'm just going to single out two organisms here, jack pine budworm and dwarf mistletoe. I'm going to talk about more on this. But, Jan Klachi, one of my PhD student candidates, just started on this mistletoe. I know I'm going to disappoint you. Dwarf mistletoe has a very soft feeling for most of you, but is a parasitic plant, unfortunately, in many plant species. They suck the resources from the other plants. At the end, I don't know, depending on the conditions, eventually they kill the trees. Even in this case, for example, tree is really dead, but looks still green. But these are not the lead needles of the pine. These are the dwarf mistletoe, as you see in here. Or this is the seeds. Let's talk about this jack pine budworm. Why is it important? Again, jack pine budworm is one of the most important defoliators in this system. And then their defoliation usually don't kill the trees, or they stress the trees. Those stressed trees will most likely be available for mountain pine beetle to colonize. And jack pine budworm become epidemic time to time. And you will find when the mountain pine beetle arrive, pure jack pine forest, might see this very large available pool of jack pine trees in the system. Therefore, understanding their interaction could be really important. Possibly, again, defoliators means that they feed on the foliage, jack pine budworm, and the bark beetles attack the stem. When you look at their biology, it's pretty much really overlap. But this is the important part, I think. When the mountain pine beetle emerges, when they're seeking the trees, suitable trees, what do? At the time when they're seeking possible potential host trees, and then jack pine budworm just finished the defoliation, and then they look for additional hosts. Around this time, all these trees basically could be highly, highly susceptible to the mountain pine beetle. I have no idea what this laptop is doing. Switching back automatically. I didn't set up anything. But this is the really important case. I want to make sure that you understand this part. This is the part that jack pine trees will be highly, highly susceptible to the mountain pine beetle attacks. So what I'm saying that pretty much their biology overlap. So pretty much, everything is suitable for their interactions. Unfortunately, there is no mountain pine beetle, jack pine budworm overlap at this stage. And we cannot really do with experiments with the live beetles. So we start using this Grossmania clavigera, that is one of the fungal associated of mountain pine beetle, to simulate the mountain pine beetle attacks and look at all those jack pine budworm, and Grossmania clavigera interactions. What is the experiment? This is what Jesse designed, basically. We'll look at it. At the initial stage, we use the jack pine seedlings in the greenhouse. We have a part 1 and part 2. Part 1, basically, seedlings defoliated with the 2 larvae or 4 larvae jack pine budworm. And then we inoculated those things, 3 fungal inoculation of 6. And the second stage, those plants in the part 1, they are inoculated with the 3 fungal inoculums. And inoculated trees were defoliated with 2 larval treatment. As a nice design, try to understand their interactions. We have a lot of results, but again, I cannot show everything. Just some of them I show it here. Since if you look at the beginning, the variations of, this is the monoterpium concentration in the tissue, and there is a huge variation between individuals. So we cannot really study directly. So what we did, we look at the change of the monoterpium concentration from this point to this point. I just want to show why we focus the monoterpians. Monoterpians is important in biology of mountain pine beetle, and actually in biology of the jack pine budworm too. That's why we just focus on here. When we look at the initial change, we didn't find really any difference here. Neither monoterpians was very similar between initial and induction stages. However, as soon as you challenge them with the different organisms, the pattern really changes. Exception with the 4L and 2L, 2 larval and 4L, treatments, all others, the monoterpian concentration went down, suggesting that they are losing their resistance. I have other more results, but I'm just going to summarize it here. In this category here, defoliation clavigera category, what we have, we've seen the delayed increase in needle monoterpenes due to defoliation to the clavigera and the fungus. We see increased resistance to the fungus. It's very interesting. We inoculate them. When we inoculate them, we see the smaller lesions. Smaller lesions, there is some speculation, but usually indicate that the plant are resisting to the development of the fungi, so they don't really grow well. We didn't find any changes in monoterpene concentration, the phloem monoterpenes. And then the other category, we have this group. The seedlings were first inoculated and then defoliated. We've seen a huge increase in needle monoterpenes. However, we didn't see any changes in body mass of the adult jackpine budworms. What did they do is very interesting. As soon as they see the changes in the needle chemistry, they ate more. We call compensatory feeding. It's very common in many insect species. And we found the monoterpenes lower. So where I'm going, this system that I explained here, not really ideal system. First of all, we didn't look at the other chemicals in the system. We have sesquiterpenes and other chemical groups, phenolics, for example. We didn't look at it. But overall, and then the simulation of mountain pine beetle attack with the fungi is not really ideal environment, but that's what we had. Well, we found that this is the basic idea. Even though we use the fungi, there is a chance that those mountain pine beetle and the jackpine budworm will influence one another by changing host tree chemistry. It's very interesting. If mountain pine beetle kill the trees where budworm larvae are also feeding, that is really bad news for jackpine budworm. They're pretty much the losers of this battle. But however, mountain pine beetles, based on other assessments that we have, is always either neutral or they're always gaining from these interactions. We also look at the defoliation intensity. Jenny did these experiments. Apparently, we see some systemic response in jack pine, but this systemic response is a threshold. Means that trees really don't care much at the lower defoliation category, but as soon as you introduce more, you see that systemic response means the whole tree defense is sort of waking up, and they become more resistant. I'm going to gear up the change to look at this role of microorganisms. As I mentioned before, the mountain pine beetle or any other beetle species are associated with a lot of microorganisms, such as fungi, bacteria, or yeasts. Among these, bacteria that a lot of people don't know, I can find out at least thousands of studies on that. A lot of people work on that. But the bacteria is really, really handful. Last 10, 15 years, some people start publishing it about the bacteria, and yeast is worse than bacteria. Only 1 or 2 labs I know they work on with the yeasts. Difficulty is we cannot identify them easily. You have to use the molecular tools, and we cannot grow them in the media. It's very difficult. But when you remove the bark of any conifer species, when attacked by mountain pine beetle, you will see this kind of discoloration. As you've seen here, these are the beetle galleries, and you see the staining. Those are fungi. And you can take the fungi, Ophiostum montium, for example, associated with the mountain pine beetle. You can grow on the pine twig. You can get this kind of thing. And these actinomycetes, these are the different bacterial group. And if you grow them in the different media, as you've seen, you get different shapes. I tell you, I enjoy looking at the insects under microscope, but as soon as I see bacteria group, I think they are more fascinating. They come from different worlds, really. This kind of shape or this kind of shape is really, really fascinating. The idea is this proposal that funded by the USDA AFRI. We're thinking that all the pattern we see at this landscape level is really reflection of what's happening on the bark. What's happening between the fungal and the bacteria associates of the beetles. How do they interact together? This is all about that. Earlier studies found that the beetles ingest or bacteria that inhibit antagonistic fungi. That's a really strong indication that bacteria has an impact on insect biology. Also, some bacteria species selectively inhibit antagonistic but not mutualistic fungi. Again, not only beetle, bacteria and the fungi in the same system, they also interact together. And then things are really complicated. Pines contain itself endophytic bacteria. They are always present on the bark that inhibit mutualistic fungi. That's the third component. And the fourth component is that tree chemicals influence all these interactions. See, my point is the system is really complex. It's not easy to tease out. But this is what Janet did, basically. She wanted to understand, okay, yes, this is from her graduation. This is her hypothesis. She tried to understand. She has these three questions. Look at the bacterial fungal associates of the mountain pine beetle will have a positive impact on beetle reproduction, regardless of host tree species. Conversely, bacteria isolated from trees and opportunistic fungi will reduce reproductive success of the beetle. And the third is the difference in the bacterial communities in jack pine versus lodgepole pine will explain difference in the beetle reproductive success between these three species, if there is any. Basically, in short, she wants to understand how bacterial fungal interaction affect the reproductive process of mountain pine beetle. And this is the project, this collaboration with Ken Rafa, my former PhD supervisor, and then Cameron Curry is a bacteriologist at the University of Wisconsin-Madison. Methodology is really simple, but requires a lot of work. She collected the beetles and identified 6 them out, male and female. She inoculated them with the bacteria. As you see, they are really swimming in the bacterial inoculum. They like it, perhaps, I don't know. And then we cut the trees. We use 10 jack pine and 10 lodgepole pine in the system. And then getting help. She did the most of the work. We also get help from, here's the Kyle, who's cutting the bark. And then we cut these pieces of the bark. We inoculate them with the fungi and created this assay unit. Instead of using whole bolts, we use this assay unit. It's very successful, actually. As you see in here, we put the beetles from here. They start tunneling, and these are the larval galleries. But you see a lot of staining. This is a really simplified system to see closer look at the beetle development and interaction with the fungi and then bacteria. And she has several variables here that relate to the beetle success. She look at the maternal gallery length, the number of larvae, larval gallery length, number of days required to reach the maximum gallery length and larval density, and many others. Okay, since we're not really bacteriologists, our colleagues from Wisconsin, they sent us these organisms here. We use 2 beetle-associated bacteria we call ABA1 and D422. And then we have also 3 associated bacteria. We say closest species is in bacteria is very difficult to identify what they are. But these are the closest species. And all of them are gram-negative bacteria associated with that. And we have also 4 fungi species here. Grossmania and Claviger and Ophiostoma montium. These are beetle-associated, and people speculated they are symbiotic. Means that they're really helpful to the beetle. Aspergillus and yellow, we call them opportunistic. Beetles don't specifically carry around with them, but they are somehow airborne, I assume. They attach to the beetle, but they also utilize the environment that's created by the beetle. But we call them antagonistic, but our results show that they may not be really antagonistic. They may not be harmful to the beetle, because we have some evidence that actually they are also helping the beetle itself. Well, I'm not going to go through all the details, but this is very simple. We have a controlled treatment, and we use the sterilized, surface sterilized insects. And this is the red one shows actually where the means are. In some cases, you will see with these brown blocks, the means are smaller than the control. And the yellow blocks show that means are bigger than the control. I don't want you to look at everything, but overall, we have some combinations, bacteria-fungal combinations are, beetles are doing very well. In other cases, beetles are not doing very well. But really, this is changes through the different stages of beetle development. And we have exactly the same thing for jack pine. I'm sorry, I cannot go into detail, but overall, and we also compared the lodgepole and jack pine. As you've seen here, some combinations are beetles are doing very well on the lodgepole pine, and others are doing very well on the jack pine. So what did we get? Well, it's really, really complex. Really complex. If you have all the treatments we have about 48 treatments. There is no way we can compare those things. So complicated. But this is the results we have simplified here. First, we have a beetle galleries were longer in assays that contain beetle or tree-associated bacteria. That's really important. Those bacteria is helping the beetle in some way. And we have a beetles with tree-associated bacteria reach their maximum gallery length faster than the beetles with beetle-associated bacteria. This is not what we expected. We expected that beetles associated with the beetle-associated bacteria, they will do a good job, but they did not. This is the experiment we have. And then we also have a 10 adults emerged from beetle-associated bacterium antagonistic fungal combination, had a larger pronotum width. This larger pronotum width indicates that beetles are big. The sizes are bigger than the others. Then the beetles emerged from beetle-associated bacterium and symbiotic fungi combination. Again, this is not what we expected. That's why we are saying that antagonistic fungi, perhaps they are not really antagonistic. They still are helping the beetle one way or another. But this is the interesting part, I think. When the beetles were with the tree-associated bacteria, they excavated longer galleries in jack pine. Okay? When the beetles were associated with the tree-associated bacteria, they excavated longer galleries in jack pine than the lodgepole pine. In contrast, the presence of beetle-associated bacteria paired with the symbiotic or antagonistic fungi, beetle constructed longer larva galleries in lodgepole pine and jack pine. Again, this is suggesting that these combinations, various combinations, have various impacts on the beetle. This is my 3 simple conclusions. First, based on what we have, I think historical interactions, evolution we call, co-evolution of the beetle, and with the tree interaction seems to be really important here. And then beetles may require its bacterial associates in order to successfully establish in jack pine. We have evidence for that. Another one that both beetle-associated bacteria and the fungi are probably providing essential subcortical environment, such as nutrients. That's not a really new news, actually. And bacteria is a news, but the fungi been reported many times that they provide nutrients for bark beetles. Probably have a lot of questions in your mind. And then the last cases I think is also fascinating. I look at the pheromone chemistry of the beetles. As we know, the bark beetles, or many insect species, they communicate through releasing some chemicals. Those chemicals that provide in communication within the same species we call pheromones. Pheromones are really important. In bark beetles, they are linked to the tree chemistry. All right. Presence of the, basically, host plant material may influence the production and release of the pheromones. I don't remember everything, but in many cases, some bark beetles may convert host plant monoterpenes. Monoterpene is one of the most abundant compound group in pines to the pheromones. But there is a recent evidence suggests that actually, they don't really need those terpenes directly. Exposure to host terpenes may also stimulate de novo synthesis of pheromones, means that they convert to pheromones very simple molecules such as sugars and amino acids. Forget those things. This is the important here. This is one of the things called hydroxylation. It's really important. Here, myrcene is one of the most abundant tree chemical, monoterpene. What did beetle? Didn't really do much. Added one hydroxyl group here and produced a pheromone. Believe me, this is a really common protocol. And here, alpha-pinene is also very abundant. Added some hydroxyl group and produced verbenol and verbenone. It's a simple changing in chemical structure. And as we know, the oxygen and hydrogen are very plenty, abundant outside. I'm going to give you the story of how this is. I'm going to put it in the beetle colonization pattern. This is the initial attack. When I say that, the interaction between the beetles and the trees really happens very quickly, I'm not lying. It happens very quickly. Everything happens suddenly. This is life and death situations. When the beetles find a suitable host, okay, they release pheromone called Trans-verbenol. Okay, that's the female pheromone. The purpose of that, bring additional males and the females to the system. Okay, that is the first stage. And Trans-verbenol is the product of the alpha-pinene. The chemically very similar. And the second day, literally the second day, if these pheromones that females released successful, what do? A lot of males will responded. They still continue producing pheromones, but arriving males also produce pheromones. It's called exobrevicomine. All right? And then this is going to bring more beetles to the tree. This is day 2. Day 3 or 5, I call it. If everything works out, there are a lot of individuals arrived. Okay, the female continue producing exobrevicomine, aggregation pheromone, but male also produce frontalin. The male produce these two pheromones. Both sexes also produce verbenol. Verbenol is anti-aggregation pheromone of mountain pine beetle. All right? This is giving a signal saying that, you know what? This host tree is already occupied, go find something else. And what do? We call switch mechanism. They attack the nearby trees. When you look at from the attack trees from the landscape, that's why you see that this wave-like mortality. This is pretty much driven from the same method. So what we do is a very simple protocol. We cut 5 lodgepole pine and jack pine. We got 1 bolts. We introduce the beetles onto them and start collecting volatiles. It's really, really simple system. We have a small pumps here, may not be visible from where you're sitting, but there are some Teflon tubes here, Teflon tubes. These Teflon tubes are attached to the individual entrance hole, but it's controlled by the timers and electricity here. And then this is the individual holes, and these are the collection vessels where whatever is coming out from this entrance hole, we trapped here. And we extract them later on and then analyze it. It's a very simple method. What we did, since the pheromone dynamics is really changes within the first 5 days, I look at the 12, 24, 36 hour, 48, 72, 96, and 120 hours of pheromones, multiple times. Results are really fascinating. I never expected to see these results, but it's really cool. What we have here, again, we have a lodgepole pine and jack pine. We have a trans-verbenol, and then as you've seen here, trans-verbenol is a female pheromone. Female produce more pheromone, about 9 times on the lodgepole pine and the jack pine. That has a significant impact. Exobrevicomine, similar. Frontalin is about 9 times, 10 times more on the lodgepole pine. This is the really interesting part. Whatever the reasons are, I have no idea. But the females or the male, they start producing anti-aggregation pheromone, high amounts from the jack pine. That is interesting. So I look at their time. Okay, this is trans-verbenol male pheromone, and this is the amount of chemicals released, pheromones released. The amounts are pretty high here compared to others. This blue line is the lodgepole pine, the red is jack pine. As you've seen here, as soon as you put the female, actually, within a couple of hours, they produce pheromone. And then after 36 hours, everything really shuts down. Here, this is the jack pine. Amount is really, really less. And exobrevicomine are very similar, but this is the pattern. Frontalin is a male-produced pheromone. As you see in here, the peak is really occurs around the 48 hours versus about 70, 72 hours in jack pine. But this is the interesting part. This is the blue line again, lodgepole pine. The beetles, both sexes, is very interesting, female and male. They produce, I didn't know that actually, they start producing anti-aggregation pheromone when they tunnel the tree. However, that production is really huge on lodgepole pine, the jack pine, compared to lodgepole pine. You see, this is several fold higher, about 6 times higher than that. That is really, really important. I never reported. I'm discussing these results in one of the pheromone chemists, retired professor, John Borden, actually. Whether this really makes sense or our error, I don't know. But these have major implications for us. First of all, since the female release less trans-verbenol, that is aggregation pheromone, if they cannot really attract male on jack pine, most likely they never attract anything else. They cannot have a successful colonization on jack pine. Plus, that's not enough, plus they're releasing high amount of verbenol, anti-aggregation pheromone. I have no idea why again. This is important because they're producing aggregation and anti-aggregation pheromone, more anti-aggregation than aggregation pheromone. This suggests that, again, colonization on the jack pine may not last longer. less long. Might seize within a couple of days. Perhaps this is really good news for the beetles in jack pine. We so worry about it, but I think this data somehow show that the jack pine and the mountain pine beetle may not be so much successful on the jack pine at all. So where I'm going from these studies, I'm going to continue focus on this invasion dynamics, but I'm going to extend it more on this. I'm going to accommodate this spatial and temporal variation in the tree chemistry. Tree chemistry changes from morning to evening. Tree chemistry changes from the north aspect versus south aspect. It's very, very dynamic. Scale, I think, is going to be very important here. Scale that processes the governing different aspects of invasion occur in different spatial and temporal scales, and their consequences also occur in distinct scales. Scale will be incorporated eventually. And then this is one of the main questions: how the climate change affect the plant defenses? We look at the plants. I know they do affect the plants. Not many people look at how the climate changes affect the plant chemistry and their consequences on the community of organisms that interacted with that, whether they will benefit or this is going to be harmful to them. Those are the things. I have a number of collaborators here. Vic is here and others, people that I collaborate at the U of A and at the University of Wisconsin-Madison, Rafa and Cameron, and Brian Alkama from University of Minnesota. I have been collaborating with this group, actually, a Swedish and Norwegian group, for the last several years. I have still doing work with my colleagues in the UC system. And we just started this work with DEPA at the Forest Service, doing a similar concept, but looking at the more spruce and balsam fir and how the pathogens affect their chemistry. We have Enrico Bonello from Ohio State University doing he's assessing our phenolic work, and then we have a colleague with Parks Canada and Susan Smart at UBC doing work with fungi. These are the funding agencies. There are several. I don't want to go through all of them, but without these funding agencies, this research won't be possible. Finally, the slide that you've been expecting. This is it. Let me know if you have any questions. All right. Are you taking your revenge right now, Evan?
That's the plan.
All right.
I should have signed off on my thesis.
All right. Sorry.
So you talked a little bit about how host switching occurs, but I'm not sure if we really know exactly why host switching occurs. Like exactly, is it because the tree is full of beetles, or do they stop? Does quorum sensing happen because the tree is packed, or does it happen because the tree is dead, effectively dead, no longer defending itself, and doesn't any more beetles to attack it? If that's the case, could it possibly be that more verbenol being produced could mean that the trees are, more weakly defended or are killed more easily?
If you look at the figures that I didn't emphasize it, but from the day one the beetle attacks, they release a lot of host terpenes. The thickness of the bar and the arrow is very thick at the beginning, and later on, the days 3 or 5, it becomes less and less. I've done some studies with that. The ratio of the pheromone to monoterpenes become really important. As soon as you increase the ratio of monoterpenes, that become more attractive in some cases. As soon as you reduce the amount of monoterpenes released from the trees, they become less attractive. I think this absolutely. This is sending a signal that tree is either occupied, but anti-aggregation pheromone is released. All beetles are released, but one beetle versus thousands of beetles, when they're releasing the verbenol, anti-aggregation pheromone, the amounts become big, right? And that's sending a strong message to the other beetle species. Yeah. But it does occur. It does occur in the beetles. So why it occurs and what is the exact mechanism is not exactly known. Yeah. Yes, Justin.
I was just wondering about the bacteria. So it seems like it doesn't matter. So the bacteria can come from the tree or be associated with the beetle, and in either case, it helps the beetle. But and you mentioned something about nutrients. What exactly does the bacteria do?
We don't know those. I'm speculating here. Exactly. We don't know how the bacteria. I don't think anyone show effect of bacteria on nutrient components of host tree species. No. In fact, there are some few studies showed on the fungi. how the fungi are affecting the nutrients of the trees. But no one really showed the bacteria. Janet, am I right?
Yes.
I think she knows literature better than I do, so I don't think anyone showed that. That is my pure speculation based on the results we have. But it needs to be demonstrated, needs to be shown. It's a good point. Yes, Evan.
So I was just wondering how, I was thinking about this in my own research, like how do you interpret larval gallery length? Like what does that mean? Does a longer larval gallery, did it mean the beetles were dying and they weren't making long galleries? Or, is a short gallery, does not need to consume a lot of phloem because the phloem is more nutritious, or is it a longer gallery than the larvae live
That's the
longer?
toughest question. I know, it's very difficult to answer that. In my view, personal view, the longer galleries indicate that beetles like the tissue and they eat it more. But alternative argument is that, you know what, they don't need to eat more. As long as the phloem is nutrients, they are full and they just lay down. They don't need anything. They don't need to excavate longer galleries, but it really changes the situation perhaps. We cannot speculate what the longer gallery means. But it's difficult. Yes?
How similar are the natural levels of the pheromone precursors in jack pine and lodgepole pine, like so alpha pinene and myrcenes? And how much variation is there within species of that expression?
First of all, there is a variation within a species itself. If you look at the lodgepole pine in North America, you will find huge variations.
Like 10 times?
Oh yeah, in some cases you will find it, yeah. The ratio, I didn't show it here, ratio of alpha pinene and other chemicals, beta pinene, they occur in stereoisomers. They have a minus and plus. That really complicates that. But basically, variation between species is usually higher than the variation within a species. Having said that, jack pine has pretty much contains all the chemicals, whatever the lodgepole pine has it, except beta-phalandrin, or very little amounts. Beta-phalandrin is one of the major compound found in the lodgepole pine, and in the jack pine is absent or very little. Jack pine is really loaded with alpha pinene and 3-carine. It's another chemical compound. It's very difficult to answer that. There is some variation you will see. But at the end, is perhaps the ratio of those compounds really important?
Have you done that experiment where you're collecting the pheromones with the
Turbines?
hybrid pines? The hybrid jack pine?
Oh, hybrid pine is messy.
Okay.
It's really messy. We did a lot of work with that, and you will go nowhere. We did bacteria work with this. Janet here, I didn't show it, that their results is really, really messy. First of all, we cannot treat the hybrid as a single species.
Right.
It's jack pine and lodgepole pine, and it changes a lot. 10% lodgepole pine versus 90%. The Francis had a presentation here a couple of weeks ago. We cannot really treat it as a single group. That's why I didn't include it here. It changes a lot. I didn't show it here. This pheromone work that I have, we also collected the monoterpenes released that. I didn't show it here. It further complicates that. Absolutely, we have those data too. It's a good point. Yes?
I was wondering, when the beetles attack the jack pine, they secrete dispersion pheromones really quickly. Would that be because it's just not a good habitat for them and they don't like it, so they're like, don't come here, it's really bad?
That is my guess, actually. Yeah, that is my guess, but I don't know. But whether that's true or not, I really don't know.
We didn't see really good success differences between beetles and jack pine and lodgepole pine, so if you had found these differences, it would have been a better explanation, I guess.
I guess so. The thing is that these results are really important for endemic cycle, not epidemic. Epidemic, I think they can kill any trees they found. They even attack the cypress trees and they kill it. It's no problem. Cypress is not one of the hosts. This is the important thing is, if you have 1,000 or 10,000 beetles are waiting for your signal there, and you're releasing even little amount, you will find some receptors there. They will attract it. They will find it and they will track it. But if you have just 10 beetles dispersed over there in the large landscape, your little signal is not going to reach them. That is the thing. Or chances are very little. That's why I'm saying that, since the female pheromone is so little, chances that they will find a suitable jack pine during the endemic cycle when the population are low is very little. Having said that, this year I'm going to test this thing actually. We found some answer student that are going to do this experiment in the field. Then I will be more confident to state, yeah, that is the case or that's not the case. yeah, it's going to be, we will be tested in the field this summer.